Hello, everyone. Thank you for tuning in to the 2020 ABE Research Poster Symposium. My name is Spencer Serrano. I'm an undergraduate student here in the ABE department, and today I would like to highlight the proteolytic degradation of alpha-synuclein by engineered Neurosin variants, which is an exciting project that I've had the pleasure of being a part of this semester. In the lab, we have two main research objectives that drive our studies. The first is to develop a highly selective Neurosin variant that can cleave a variety of alpha-synuclein proteoforms and prevent their aggregation. The second is to determine if an engineered Neurosin variant can be used as a therapeutic for synucleinopathies. Synucleinopathies are a group of neurodegenerative disorders associated with the accumulation and aggregation of the alpha-synuclein protein, and it includes Parkinson's disease, dementia with Lewy bodies, and multiple system atrophies. As you may know, these disorders are extremely prolific and are estimated to affect 5 million people worldwide. Currently, there are no effective treatments. Neurosin is a great enzyme for our studies because wild-type neurosin is a protease that is naturally expressed in the brain where synucleinopathies occur. Additionally, neurosin has been demonstrated to cleave alpha-synuclein and prevent its aggregation. There are some limitations of the wild-type enzyme, however. It is capable of cleaving a multitude of other unrelated substrates, which can be detrimental to the treatment. It is not efficient at cleaving alpha-synuclein mutants, which are also associated with synucleinopathies. The principle that we use to design our enzyme is called directed evolution. It is a technique used in protein engineering that allows us to design and customize proteins through a process that mimics natural selection in a laboratory setting. Directed evolution consists of iterative rounds of mutagenesis, followed by selection or screening, and then amplification. Our specific directed evolution platform is referred to as YESS, or the Yeast ER Sequestration Screening System. Figure 1 shows the YESS platform, in which a protease, in our case, neurosin, is co-expressed in yeast cells along with multiple substrates. When the protease cleaves a protein substrate, an epitope tag is released, and subsequently, a fluorescing antibody binds to that tag, and it allows for FACS screening. Figure 2 is a closer look at the YESS platform. The top sequence is responsible for the expression of the protease and contains a library of Neurosin variants for us to examine. The bottom sequence is responsible for the expression of the substrates. The first substrate, AFRFS, is a counter-selection substrate. And it has been demonstrated that neurosin has a high affinity for this substrate. It is attached to an epitope tag. The second substrate is the selection substrate, VAQTVEG. And this sequence is within the alpha-synuclein protein in a region that is responsible for this aggregation. So through this system, we are able to identify Neurosin variants that have a high affinity for the VAQTVEG substrate as opposed to the AFRFS substrate. And we are able to identify Neurosin variants that might be good candidates for our enzyme. Figure 3 shows our results. And you can see that there are 7 different Neurosin variants that have a high preference for the selection substrate over the preferred substrate or the counter selection substrate. And this graph is normalized to the affinity of the wild type Neurosin enzyme. So through the YESS platform in the directed evolution process, the Denard lab has identified 7 promising Neurosin variants. Proteolytic degradation of alpha-synuclein has a potential to significantly transform how disorders like Parkinson's disease and multiple system atrophy are understood and treated. Designing fast and selective Neurosin variants is among some of the first steps to bringing the proteolytic treatment of synucleinopathies into reality. Upcoming work that we're going to do in the lab consists of characterizing the engineered Neurosin variants on the alpha-synuclein substrate We will use internally quenched fluorogenic peptides with a selection substrate and the counter selection substrate. And this assay will allow us to determine the kinetic parameters of each neurosin variant. And these kinetic parameters will guide us through the future engineering of our custom protease. And the second objective is aiming to deliver neurosin variants to synucleinopathy mouse models, which will show us how neurosin variants behave in the brain. Thank you for tuning into my presentation.